This podcast is brought to you by Bionomous, bringing automation to the laboratory, and brought to you by the blowfish. The blowfish is able to blow itself up much like a balloon, except this balloon isn't released by a poor crying child eating ice cream. They inflate themselves with water, becoming caught in the throats of their predators. Impressive feats, my dear blowfish.
Hello, my fishing friends, and welcome to another episode of the Getting Fishy With It podcast. I'm Josh.
I'm Amber.
I'm Christine.
And today we have a special guest from the company Bionomous. Welcome, Anna.
Hi everybody.
Hello, welcome. We're so happy to have you.
Yes, we are. And before we get into what we're going to talk about today in Anna's company, we're going to quickly just go over how everyone's week has been and what's new with everyone. So, Amber, would you like to start?
Yeah, so something exciting has happened for my institution at Northwestern. So we just recently went remote. Or primarily remote. So still like working within Illinois, but very exciting as we get to work from home now. And occasionally we'll go on site. Yeah, most of the time. I would say like 90% of the time. But we will go back on campus to do like inspections since I'm part of like the IACOOK team or department. And so we'll go back to do inspections, have like our get-togethers and things like that. So we'll still, meet up every once in a while. But otherwise, yeah, just like chilling at home with my puppies. They love it so much.
Of course they do.
That's so great.
Yeah. And not having to battle the commute every day because I used to take the train, but it's gotten crazy. So very excited.
I've taken that train in Chicago and I was worried it was going to fall off the tracks or the tracks were going to fall down.
We're doomed.
Doomed! Doomed!
So
oh no. Yeah. Yeah. Well, hopefully they improve that infrastructure in the coming years.
Hopefully.
Yeah. I'm so happy for you. I wish that someone could develop some technology so I could remotely feed my fish so I could work from home as well.
Yeah. What about you, Christine?
I think I have just one exciting thing. It's been a bit of a busy couple of weeks, just facility operation wise, but I just got accepted as a volunteer for our local zoological facility. So I'm going to my first orientation and everything this weekend. So I can't wait. I don't even care if I have to scoop poop all day.
Well, we do get $1.35 a day, right? Best job I ever had. Best job I ever had. Best job I ever had.
I am fine with that. Or cut vegetables, even if it is for great apes. I don't care. I'll do it.
Oh, I love this.
I won't do it with a smile on my face, because great apes are not my favorite. But no, I'm just excited for, just kind of a new opportunity and get to see a little bit behind the scenes at the zoo, which is cool. How about you, Josh?
Yeah, I just moved this weekend to Yonkers. So, and the thing that I'm most excited about is we got this giant sectional couch and it's the most amazing thing I've ever seen. It's incredible. Like I don't even have words. Like we lie on it and we're just so happy and we just look at the TV. So like we finally got the TV set up. I got my computer set up. So like life is really good. And like we decided we're going to be extra organized. So we're going to do like, have you ever seen those YouTube organization commercials?
Yes.
Like the YouTube organization videos? It's like ASMR where they like put all the toothbrushes next to each other and everything. Like that's going to be our lives now.
You wish to be Amber, like to stay at home all the time now, right?
Yeah, exactly.
Did you get that sectional from Costco? Because I've seen that sectional at Costco and like I have coveted it.
It's mine. My precious.
I have to say.
No.
Oh, okay.
We went to Bob's Discount Furniture. I don't know.
And
we were like, do you have any deals? And they were like, the deal is it's Bob's.
Well, this sucks.
It's already discounted. And I was like, okay, dude.
Oh, wow.
What do you want? This is New York or New Jersey. You get what you pay for.
Yeah,
Cool.
so that's it with me. And once again, we want to introduce Anna Hernando. Well, introduce yourself.
So I'm the COO and co-founder of Bionomous or Bionomous. I don't know how to pronounce it. Depends on which language. Thank you very much for actually inviting me here. I'm super excited. So a lot of new things. This podcast is new in my life. Participating in a podcast, so it's actually great and challenging also, I would say. But it's amazing. So thank you very much. And then a lot of new things this week. Is my first time in the US.
Yeah.
So it's been like super cool, learning a lot about the American culture, also like a lot of new talks, new people, like new cities, like I've visited Boston and New York now. So it's been like quite challenging and like very exciting week.
That is so cool. I personally really love Boston, so it's very cool.
I told them we're going, by the way. Yeah? I told them we're going to PAX. Yeah.
Yeah. That's, I guess the other thing that's new with all of us is that we're giving a panel at a pretty big convention. So that's very exciting. But I hope you had a good time in Boston, and hopefully New York is great too. So that's awesome. We're so happy to have you. Shall we get into the episode?
Bring it on.
All righty. So Anna, do you want to talk a little bit about your background in the field and kind of how you got to where you are today with this company?
Yeah, sure. So basically, myself, I'm a biologist by training. I did my degree in Madrid, and then I moved to Switzerland to do my master's in medical biology. So, you know how in biology we have like people that are boat or lab, or at least we say that in Spanish, like bata bota. And then I took the lab coats one. So I did molecular and cellular biology and also a bit of neurobiology. And then when I was in Switzerland doing my master's, then basically I got to know Frank, who was doing this amazing project, which was to develop at that time was a prototype. At some point I have to show pictures of how the first prototype of the EggSorter looked like, because but he was developing this prototype to sort fish eggs. And then this is how I got in contact with zebrafish. And basically since then, it's been 4 or 5 years working with zebrafish and learning a lot about the model and how it's been used in science. So that's a bit my background.
Very cool. Did you have, before you kind of took the science route that you did you have any kind of experience working with animals or fish or any sort of species beforehand? Because we find generally that a lot of us that end up working in these fields, we're all kind of animal nerds from the start, or we have a really serious interest in biology. So how about you?
So me, I had some experience with mice, but I like it much, to be honest. I was a bit, not traumatized, but it was hard to work with them. So then I decided I will try with cells. So I was working in a lab that was working with cells as a model, with neurons. So then that was much better, I have to say. So then basically with zebrafish, I did not have much experience until I got in touch with the EggSorter. And then when Bionomous started, I remember one day I saw somebody in my lab working with zebrafish. I was like, oh, that seems cool. Can I get a tour in the lab? Cool. Get to see the facility. And the girl was doing micro injections. So it was my first touch with the zebrafish. Then nothing else. And then until Bionomous came to play. And then I was like, whoa, this model seems cool. And then this is how all started, and how I became, I would say, a fish nerd now, maybe.
Yeah. We induct you as a fish nerd. We are we're the council that decides those things. So you are officially you are a fish
by
nerd.
the end of the podcast, then you will approve me as a
you're already approved. You're already approved. You're good to go. But yeah, no, that's really cool. I think I know a couple of colleagues of mine that didn't do anything with fish before they started in the fish labs, and they kind of came on as an undergrad and they're like, I want to do this. So that's very cool. That's very cool. Thank you for sharing that. I'd like to talk a little bit about Bionomous. Can you tell us a little bit about when it was founded and all the kind of stuff about where you guys are based? Just a little bit about the company itself.
So we started 4 years and a half ago. The project had been there since 2017. And then basically, it all started as an idea that Frank had, because he was developing this small, actually it's a fun story, I would say. He was developing these 3 like not 3D, but like a robotic fish that was mimicking fish behavior.
Cool.
So it was like a 3D silverfish that will just move around, and then they will put them in an arena with other fish and see how the fish interacted with the robot.
Very cool.
And then basically, so he saw that the people were spending a lot of time just sorting fish eggs. He's an engineer, so he was like, oh, I will be a robot for that too, like so that you don't have to spend the time hands-on, like doing these mechanical tasks, so that you can have more free time to just do the experiments with the fish, actually. And then the project started in 2017, and then I joined the team later on when we were looking for other applications of this technology, so other fish species, other organisms, like other models. And then after some months working in the project, we were like, oh, this is so cool. We have to really put it in the market and make this available for all the researchers. It cannot stay in the lab, but it has to go out, which is a bit scary. It's like putting a baby into the world. So yeah, it's it's like, oh, are we sure about that?
Make me proud.
Exactly. And then, so we started little by little. So we started at the EPFL. So we're a spin-off of the EPFL in Lausanne, Switzerland. And then since we started, it's fun to see how we started like in a room with 2 desks. At some point, we were able to fit like 6 people there in 2 desks. So that was challenging. And then luckily now we're at a bigger space. So we moved to the countryside in Switzerland. I would like to say there are more fish than people, but it's not true. There are more cows than people.
Right.
So,
Definitely. Yeah.
But it's a beautiful place. So this is where we design the technology and we assemble the machines so that they are ready to be installed in the labs.
Very cool. That's very cool. Anybody else have any questions about the company itself or anything?
Yeah, I guess like what was it sort of like what drew you to? So did they recruit you or you found them? Like how did that how did you find Frank, I guess?
I mean, if I'm completely honest, like being a student in Switzerland is not the easiest thing on earth because everything is super expensive. So I was just like, oh, I want to be honest, like get out of the lab, like try something that is not much research. And then I was looking for a job, and then I saw this job ad that was like market research for something related to biology. I was like, oh, this is super cool, because actually it was a great match, because it's like Frank brings this engineering background to create the machines and I bring the biological point of view. So it's kind of actually the name Bionomous, it's that, like you have bio from biology and nomos from autonomous, which is the engineering part. So
very cool.
We build machines for biology. So it's like a match. It was a match there.
That's
we
cool.
love some good wordplay. I feel like that's a good matching of words.
Absolutely. So. Yeah, that's so great. Very cool. Any other questions?
Yeah, I would just like to hear more about your like day in the life as like a COO and co-founder. I think that's just like an amazing thing, like especially for, you know, a woman such as yourself. So just hearing more about that.
Well, thank you. I have to say it sounds super fancy. It's like when you're in a startup, in the end, it's like you basically have to do all the roles possible. Like you have like all the hats, and you have to be switching from one to the other one. So at least at the very beginning, it's not a very defined role. It's like you do what needs to be done, and then you learn all the possible things to be learned. Like basically you're switching from one thing, like from, okay, like now I have to deal with having the fish, or now I have to deal with, I have to install a machine, or now I have to deal, we need to have this person in the team.
It's so impressive that, you know, it's so difficult to wear so many hats, like you mentioned, just to have this role where, you know, you are doing your main role in the title, but you also have to do so many other things. So that is super impressive. So thank you for sharing. That was great. Next, I would really like to chat with you about the EggSorter. Do you think you could share a little bit about, you know, what the product is? What does it do? You know, kind of what was the design behind this planning?
I think it's good you ask me that, but because basically, as I say, like the EggSort is like now like our little baby. So it's like, when you have a baby or kids or dogs or puppies, fish, you always want to show pictures and talk about them.
Absolutely.
Fish.
Yes.
Yeah.
So he's like our little baby fish.
I wanted to show you guys a picture of my sectional couch. That was what I was the most proud of.
Don't show anybody pictures of your aquarium, Josh.
Hey. I moved and I fixed it. Okay. It looks very beautiful now, but I'm still afraid.
You sure about that?
I'm afraid to show.
I'll be the judge of that. I want to see the photos. I need photo evidence. I don't believe you, but okay.
Okay, sorry, Anna. I derailed us with my couch talk.
No, it's totally fine. At some point, I think you can buy like a small couch and put it in the aquarium, like fish will like that.
And then a tiny fish tank next to that couch inside the fish tank?
Yeah, do it. You need to get a sectional design that's identical to the one that you have in your house. That's what you need to do.
We need enrichment.
We need to make this work. We need to figure this out. Someone with a 3D printer and just do it.
Be a loop.
Exactly.
Loop. Yeah. All right, sorry, I got us off topic. But please, let's talk about the EggSorter. What's so much more important than my couch?
But yeah, it all started like actually it was also a 3D printed part at the very beginning. Like really, we have to find. It was like a 3D funnel, like leading to this small wheel. It was like 20 centimeters. Sorry, I don't know how to translate that to inches.
I'm good with centimeters.
You're good. Yeah, this is a science podcast.
Yeah.
So it should be good. And then, from there, like basically we did like a lot of prototyping during these past years. And then last year we launched to the market the commercial product. And basically the EggSorter is what its name says. So it's a machine to sort eggs. So we started originally with the zebrafish model, and how it works is like the idea is that we have this device that it's portable and actually it's like a desktop computer size. So it's the size of a desktop computer, so it doesn't take all the space in your lab. And the idea is like you plug it in and it's ready to go, and it's user friendly and very intuitive to use. So you will have one entry for the eggs, and then the eggs will be guided into this wheel that will take the eggs one by one and put the eggs in front of a camera. So then we take one picture per egg that is then analyzed by an AI algorithm. So this is super cool because, we can talk about that later, but it can be adapted to different projects in research, different like trying to find different visual cues or morphological features in the eggs. And then based on this classification, then the eggs will be sorted into different output containers physically. So you can say like, I want to dispense my good eggs in a 96 well plate or in a Petri dish, different sizes. So it's quite customizable at that point. So then, basically we started for zebrafish. And now the cool thing is this technology can be adapted to other fish models like killifish, Neraka. Even we can also talk about this later, like flower seeds. So expanding this technology to other models, open to discuss if you have any crazy ideas. We love crazy ideas. So any crazy models that you might want to put inside the machine, we will be open to try them.
So I just wanted to elaborate on one thing, and please correct me if I am wrong on this at all. But as someone who is a person who works in the fish mines on regular, so I am in that fish room all day, every day. One of the roles that I have or that I assign to someone is, we'll produce a whole lot of wild type embryos. So we'll make a whole bunch of zebrafish wild types. One of my roles as facility manager is to maintain some of our background lines. So our wild types, our Caspers, etc. And I can make thousands of eggs. Well, I don't make them. The fish are making them. I'm not taking credit for that. Let's not get into that territory. But, my fish charges will make thousands or tens of thousands of eggs in a week. And then some poor sap has to go through and sort those eggs.
This is the worst.
So they are literally using their human hands and their human eyes and the maximum speed that they can do as a human to sort these eggs, over multiple days potentially, to try to find the eggs that are good or not. The other thing that's of particular importance for us, because we've been burned before and other labs have been burned before, is looking for screening those eggs to make sure they don't fluoresce.
Yeah.
Because that's a great one.
I always screen my wild types because I have been burned in the past. That is a story for another episode. But, given fish or eggs that are ostensibly ABs or whatever wild types, and then you look at them under the fluorescent scope and they are blue. And it's like, what? Why? So, I mean, ostensibly, the EggSorter is precisely to do this kind of labor, right? Like you are using this device and the tools that it uses on board to sort embryos, correct? Or eggs, particularly.
Yeah, exactly. So that's totally the initial goal of the machine. This is how the whole idea started. So by watching like people like you, like that your time is valuable just doing this screening in a microscope by hand. So it's like, I bet you have better things to do with your time than being in front of the microscope. And more interesting, I would say.
Yes.
And also because at some point, like you get tired and it's like you've been looking at eggs for hours. So then at some point I think like you might even dream with the eggs, which is not something that we love to do. But then, it all came from there. Like, okay, we can make these people life easier and more valuable by doing this automatically. So these mechanical tasks will be done automatically. And that's the goal. It's like at the very beginning was like just to sort fertilized versus unfertilized. So the machine has been trained and has this algorithm embedded in the device in the sense that it, like from very early stages, like even like 2 hours post fertilization, even before, like we have some images of like 2 cells, embryos, 4 cells, embryos, and the machine is able to distinguish those viable embryos for unfertilized or dead ones. So it's a very repetitive task and really, it's like mechanical task that then the machine will do. And then basically you will just put your thousands of eggs and then you will just click on play and the machine will do the sorting for you so that you can go away. And then actually something cool is like you don't even have to be in front of the machine because the machine will send you an email when it's done. So you can just go and do something else, like not even need to be in the same room. So that's the original goal. Yeah, so maybe like regarding the second point that you had about like how you need to screen for like, checking like your eggs are non-fluorescent. I think it's funny because it's like normally we get the opposite from researchers. They want to screen for the fluorescent eggs. So they really want to pick up the ones that glow. So it's an interesting point of view. It's just the other way around. But actually like the machine can screen for fluorescence too. So you will come with your 1,000 of eggs and it will pick up the ones that are fluorescent and you can tell the machine like, I want you to discard these eggs for me because I don't want to be doing the picking up. So this is very interesting when you have, for example, in your case, like thousands of eggs and there will be like only a couple of eggs that will be fluorescent. So you need to spend like a lot of hours in the dark room for just picking up like, I don't know, 100 out of 2,000 or like 10,000 eggs. And then you'll be like, okay, it's not what I would like to do this morning.
Sure. Yeah. I have absolutely spent a couple hours in a day at a scope trying to check all the different colors of fluorescence, for my eggs for that. And so for me, like I am not making transgenics. I'm not making mutant fish. I am simply trying to maintain quality, non-contaminated wild type lines. And, I don't know if Josh, if you want to elaborate on why it's important to be able to screen or not screen for fluorescence potentially.
I'm going to say 2 things actually. So number one, sort of the way that Anna is used to having people screen, I do think there's like some huge usability there. It's very important because, I've done quite a lot of fluorescent screening in my life, and there are things that get very annoying. And one in particular is like when you go into that dark room and you turn on the light unit, you're going to see fluorescing from the fish that you want, but oftentimes the ones that are not fluorescing, you can't see them very well.
Right.
So you have to be very careful. Like, so I usually find myself having to like prod the positive, like let's say a positive GFP fish, like whatever's labeled, prodding it away from the other fish and then pulling it up just to make sure that I'm only collecting that fish and not collecting the negatives. And it takes a long time. So it's you can easily put a few hours in on just a few petri dishes if you're looking around. And then also like, just I guess to the second point, as I was going to say before, I think the importance of not contaminating your lines is just that when we say the word contamination, we just mean that, these fish can be labeled a certain way. Whatever the labeling is, that's synonymous with whatever tissue or whatever, maybe mutation you have or something like that. And so what you don't want is a certain tissue type to be labeled a certain way, and then that's not what you were looking for because you were labeling another tissue type, right? And so let's say that you're looking at a certain valve in the heart or whatever, and then all of a sudden the other side of the atrium or something is there's a problem in the outflow tract or whatever it is. If there's multiple things that you're labeling and you just want to label these particular subset of cells, you're going to have some real issues later on if that gets cross-contaminated, right? So that's the reason why you want to avoid that. And this is a great way to make sure that you're selecting the right fish or selecting out the ones that are glowing so that you're only using your wild types. Absolutely.
And I mean, as someone who has assigned folks to sort eggs for me, the poor people that end up having that role, whether you're sorting eggs for, fertilized or unfertilized, or you're even sorting for fluorescence, there's no way around it. It's an ergonomic nightmare. So, just having to have someone sit there hunched over, counting, or in the dark, just try, and then your back hurts afterwards. And it's just, as Anna said, there are a million things I could be doing that would be more beneficial, that would not take up my time, sitting and sorting eggs, picking out dead ones, picking out, screening my fish. There's a million things I could be doing that, so, and I think probably, Amber, you can probably chime in on this, but from, say, an IACUC perspective, or even 3Rs, being able to sort and potentially, destroy eggs before they hatch, if they are not what you are looking for, is potentially a bonus, from an IACUC perspective, where we don't, our NIH assurance that animals need to be counted at, hatching. I don't know if you have anything you want to add on that end of things.
Yeah. And so there's been, I guess, recent legislation that has come out to where institutions now need to count embryos or count animals the day that they are born. There's still some kind of talk surrounding that, just because for a lot of PIs, especially if they're looking at the larval stage, they're not going to use those fish past the 7 DPF or days post fertilization. And so that can be really problematic, especially if you have thousands of embryos that you have to go through. And so I could see this really being a good way to kind of cut down on the amount of embryos that the PI needs to utilize, because if they have an excess amount of fish that they don't want, just because, with ergonomically, like Christine was saying, it's really taxing. And so you could miss hundreds here and there, especially if you have thousands of embryos. And then those end up on the system. And then, CCM or the animal care staff have to go through and feed those fish. And they're just basically growing up and not being utilized. And so to that benefit, I think that would be, both beneficial for IACUC, just to make sure that we're using a good amount of fish for these studies. And then also for animal care staff and for the users themselves.
For sure. And I think, I couldn't count on two hands the number of times where, a task that could be automated, like sorting eggs for various characteristics, gets missed because there's not enough time or not enough people or resources in the lab, human resources in the lab to do it. So then they're I'll just grow them and we'll figure it out after the fact.
Yeah.
Whereas, this is something that could be automated and is being automated by this device, always being improved upon. And it just, to me, seems like it's definitely a route to reduce, animal waste, that kind of thing. Yeah. Not trying to be presumptuous, but just as someone who manages a facility, that's just something that I foresee, when I'm looking at our inputs as far as users bringing me animals that may or may not could have been sorted before they ended up in my incubator at 5 days. So, any thoughts on that, Anna, or anything else you'd like to add?
Yeah, maybe something, it's been very interesting, we've been discussing with Chris Lawrence recently, and basically a point that he's mentioned is regarding like 3Rs and also refinement, like taking care of the animals, it's like all this time that it's being used for sorting eggs could be used for taking care of the animals.
100%. Absolutely.
Which is something that sometimes, because like priorities or like because like time is scarce, then people don't tend to do, or like it's something that, oh, I will take a look afterwards, or like maybe not with as much care as it should be. Then all this time could be valuable to say, hey, actually I have a couple of extra hours. I will just see how my fish are doing.
I am biased, but for me, if I am counting a whole bunch of embryos or a whole bunch of eggs, after I'm done, I just want to go home. I don't want to see any more fish after I do that. You know, if I'm sorting 10,000 into dishes of like 30 or 50 animals. So on that topic, like the egg sorter it sorts the eggs, and then you can set it up to kind of decant those eggs into different vessels. Like, can you do petri dishes? Can you do like a conical tubes? Like, where do the eggs end up after they've been sorted?
Yeah, so they can be sorted into different containers. So you can, like, the user can really choose where they want to go. They want the eggs to go. So basically you have like this part where we have 2 Falcon tubes, which is like for the bad eggs, the eggs that you would like to discard. And then the other one for the good eggs. So that is the fastest way of doing it. But then the cool thing is you can also plate them in Petri dish. So you have space for 4 different Petri dish, or like multi-well plates. So you can actually dispense them in 96 multi-well plates, which I don't know if you're doing like toxicity studies, might be like super handy to already plate the eggs directly, because I think that it's a nightmare also from what I've heard to plate the fish in the multi-well plates. But then for facilities, I guess for you, like the fact that you can set, I want like in each Petri dish like 30 or 50 eggs, and then you will just tell the machine that, and then the machine will plate the Petri dish and it will tell you when they are ready, and then you can change them with new Petri dishes like super fast, less than a minute, and they like continue go on. I just want you to plate again like another 4 Petri dishes, and really you can choose how many eggs per dish you want. You can choose the amount of water that you want. Like, it's really up to the user. So
that's great. Like from one of my tasks in my facility is I am putting the animals at 5-day post-fertilization approximately on the nursery. So we run the nursery for the users. And one of our biggest challenges is ensuring that the users actually adhere to our guidelines for how many embryos are in each dish. So, you know, automating that and having a system where it's counting for you would help me a lot, because I don't have to chase after people and say, there's too many in here and I'm not writing another label. You do it. Please don't make me write another label. You don't want to have to read my writing, please.
Christine, have you ever handled like babies at that? Like, they're so small, and I'm just like, please don't make me like put them into a different tank, because like they're so tiny and so fragile. And I'm just like, but sometimes you have to, especially when, you know, like we're using our best judgment, but it really may not be the best judgment.
There is definitely a time between like 3 days post-fertilization and 7-ish where they can get really difficult to manipulate, to try to move them without chewing them up in a pipette or something. So absolutely. So if for me, and when I'm training my staff on doing this manually, I'm always having them do as many of the manipulations with the larvae when they are still in the chorion as much as possible, because it's so much easier and safer to move them and manipulate them at that point when we're talking about, you know, animals we're growing out on the system for additional broodstock or whatever. So
maybe here to comment that, because I'm thinking about the 3Rs and so on, it's like all these eggs that are actually used for training people, like the fact that you will have a machine, you don't need to train all these people for that long, so that they're able to actually know how to pipette the embryos, because they will be pipetted by the machine. So absolutely, in the end, it's also the training, it's like much faster. So you have like, you save time there, you save resources. So that's one of the cool things too, that it standardizes the things. Like
absolutely,
like, 50. That's it.
Standardization in our line of work is really, really important. And so that, I think most labs would probably agree with me that one of the biggest challenges that they end up seeing is, you know, a lot of the time if you're manually sorting things, it depends on the skill set of the person doing that sorting, right? So I have definitely trained folks before where I will give them instructions, and again, it's time I could spend doing something else. Showing them how to look for dead versus alive, etc., fertilized, unfertilized. And then I'll go back and look at the dish and I'm like, do it again, please.
Oh, no.
It's happened a few times. It doesn't happen every time, but sometimes it's a matter of even someone's expertise or like past experience with fish and like just in general. I know you guys have all experienced this. Some people are just squeamish about animal stuff. And they're hesitant to like sort things a certain way when it comes to the zebrafish eggs because they don't want to kill them or hurt them, and they're afraid, because they just don't have that expertise. And I mean, something like the EggSorter would eliminate that from, the challenge that we have with this pretty monotonous, pretty like automatable task, right? So
I have a question really quickly. I don't know if you touched on this, Hannah, but what age can you use the EggSorter up until like what age can you use the embryo? Basically, I just want to get a sense of.
Yeah, but I think it's a very good question also because we were mentioning before the larvae. So the EggSorter, it's for eggs. So we can work with the embryos until they hatch. So I would say the limit is between, like 36, 40 hours post-fertilization, because then at some point the chorion gets like super fragile. So it goes from laying until like this age when they hatch.
Yeah. And so I personally have only had experience with like zebrafish embryos, but we are curious to know if there's other uses for it, such as for like Medaka or killifish. So if the EggSorter could be used on those. And then Josh can probably talk more about this, but like about seed tech. Not really sure what that means, but
oh, I just read it. I read it on the website and I was like, I need to know more about seed tech. That was
yeah, it'd be cool to hear what other species or what other kind of species might be on the horizon or that you're working with right now as far as other egg-bearing species that are important in lab animal medicine.
Maybe just because I'm very curious, like Josh, why is it that you're so curious about the seed tech? Like, because you have a garden and then you you grow your flowers, you want to do it in a more efficient way? Or
it's weird. I think it's just because I just didn't expect it that I just want to know more about the seed. Like, it's just not what I would have thought. And so I just thought that was just so out of left field. I was like, well, I need to know more about the seeds. I wish I had a garden. The sectional doesn't have an area for the garden yet, but maybe someday.
Maybe next version.
Yes.
So actually, I mean, now we've broadened the application for the EggSorter. So we started with the zebrafish, and then we were like, okay, this is very cool, but this can actually help a bunch of other people. So basically, we did this market study and tried to figure out which other fields this technology could be useful for, because the goal is like this technology can help as many people as possible. And then we were reading about different models, and we had some crazy ideas about Drosophila or C. elegans. They didn't work out. I have to say, they were super small, super different. But then, we tried with other fish species. So we came to work with Medaka eggs, killifish eggs. So actually, it's very interesting because at the very beginning, we thought it was going to be easier to implement this technology for these species because, like the eggs, they are the same size, they are transparent. And then, this is always funny when we try new things. So it's basically, we just take the eggs, we put them inside the machine and we're like, let's see what happens. And I remember first time we tried with killifish, the machine exploded. I mean, not exploded.
The eggs get like sticky, right? Some of those species have kind of sticky eggs. So I don't know if that impacts the ability for it to sort or...
So, I mean, I have to say it did not explode like a bomb.
Sure,
but basically, there was this tube that just went crazy. And it's because these eggs, they are super rigid compared to zebrafish eggs. So actually, it's very interesting to see how even with the nail, like you cannot break them. So, of course, our system was not thought for those kind of eggs. And then we had to figure out a way on how to work with those eggs so that the machine will not explode again, right?
Sure.
So then we figured out a way, and then now we can use, because Medaka is the same, they're like super strong eggs and they're sticky, so the physical properties are totally different. So then now we have this system that can work with different types of eggs, like soft, rigid, also opaque. So this is where the seeds come from. Like we have actually like the possibility to add a second camera so that we can image both sides of the entity. We have like different kind of illuminations, like for transparent entities, for opaque entities. And then, like seeds, they were actually easier than expected. Like in a sense, we're like, oh, let's try to see how it goes. And the funny story here is like when we placed poppy seeds inside the machine and we were like, let's try the fluorescence here. We discovered that poppy seeds are actually autofluorescents by nature. So then we had this, it was crazy, this amazing image of a poppy seed that looked like an alien. And we're like, oh, that's so funny. So now we're really open to try new fish species, new organisms, new entities, because we've learned quite a lot in this way. So now we're like also faster in the way of adapting our technology to other models. So really, if you have new ideas, really open to discuss about them. So sorry, I was curious to learn what are the species that you have in your facilities. Like, do you also work? I know Josh works with killifish quite a bit, but do you work with other species in your facilities?
So for me, we're all zebrafish all the time, baby. But that is like my current job. My previous job, we worked with lots of different species. So we had salmonids, so a number of different species of salmon and trout. We also had a number of different species of tropical fish, so things like polypterus and a few different species, and we bred those. We also worked with Xenopus and various other like frog species, the Lavis and the Tropicalis. So the Tropicalis are the little mini versions. We worked with all those different species for sure. But and I think you see, I don't know, maybe I'm wrong, but more often than not, facilities will have an aquatics core where you have zebrafish for sure, and they may be kind of the bread and butter, but then they've got a whole bunch of other species. Or as I see in my facility, folks are trying to branch out from zebrafish to other model organisms that are also aquatic. So, and I'm like, cool, but not in the same fish room, please. So, yeah. How about you, Amber?
Yeah. And so we mainly have our like zebrafish facility, but we also do have frogs, which I feel like those are kind of really clumpy. So and a lot bigger actually. But we also do have lampreys too that we usually get like during the summers. And so, I feel like working with lamprey eggs would be pretty cool.
Yeah, like the frog world is actually a very interesting world. So we've been trying to adapt to Xenopus oocytes and embryos. It's work in progress actually, because it's a bit more challenging than expected, because like the eggs are also like softer and more fragile than fish species or the fish species we work with. But interesting.
Yeah, for sure. I'm going to go find a poppy seed bagel and look at it under my fluorescent scope at some point. Have a look at the seeds. And I'll run out and see what else fluoresces.
Scraping things off the ground. I'm doing it for science.
Absolutely.
Leave me alone.
One thing I did want to chat about a little bit more, and I think this is a topic that's like a hot topic for a lot of folks, is the role that artificial intelligence or AI plays in a product like the EggSorter. Can you chat a little bit about, what how AI works for the EggSorter, or, the kind of methodology that's used, and, just kind of anything about how you've been working with AI to develop the tools that this uses?
Sure. So basically, our AI is image-based, because we take these images of the entities, and then it's really easy for us to build an algorithm that then we classify. I mean, we need a data set that has been labeled by humans. So we need humans to tell the machine what they want to sort for, like this is a good egg for me, this is a bad egg. And then we will need like a couple hundreds of labeled images per category. So, I mean, I don't know, for example, we can imagine like have this morphological feature that I'm looking for, and like I can see it in this bunch of images and I cannot see it in this bunch of images. So then you will give us this labeled data set, and then we will just train an algorithm that we will implement directly in the machine. And actually, this is how we've been working so far. But now something that we are developing and that it's in beta mode is like to give the user the possibility to create their own algorithms. So to say like the user will take the images from the machine and we'll have this, we call it like the Tinder app for eggs.
Right.
Swipe right, swipe,
So it's
swipe
like
left.
training the AI on your own data set kind of thing.
Yeah.
Okay.
Exactly. Yeah. So you will get the images from the machine and you will swipe right or left, depending if it's good or bad. And then in the same application, you will be able to train like there's no need of knowing how to code, because I mean, I'm a biologist, and to be honest, for me, like AI, it's like more like a black box.
Sure.
So, so I don't know how to code. And then the cool thing is like the engineers were like, no worries, we will create something for you, Anna. And then they created this app where I just have to click like train, and that's
Nice.
it. Like then the app runs the algorithm, and then it will give me an algorithm that I can just play back in the EggSorter to do the sorting for me. So like this, also the data stays in your facility, and you have total control on how you want to train your algorithms and do the classification. And gives also like the freedom to experiment with it. Like kind of, we have the basic application like fertilize and unfertilized, but with this AI training, you can be like, oh, I want to sort based on this fluorescent pattern because I want to look the eggs that express fluorescence in this specific line. So then you can train an algorithm for that. Or as I say, like you're looking for morphological features. So then you will be able to also like train an algorithm for that with the freedom of doing it whenever you want and whatever you can, basically.
That is super cool. I can't even, when it comes to things like that, I can't even begin to think of all the like opportunities that would, bring forward for folks that are looking to screen for very specific, expressions of a phenotype or whatever, so that's very, very cool. I love that idea, the Tinder for Tinder for eggs.
Tinder for eggs. That's gotta
so
go
great.
somewhere.
Yeah.
I love it.
That would be in the marketing where you'd have. Yeah, exactly. It's so great. So speaking of marketing and media and such, did you have anything you want to share as far as, media attention for the EggSorter, or, institutions that maybe have things to share, anything like that?
Basically, I mean, we're trying to communicate as much as possible as we can about science and the science that it's being done with zebrafish. So actually, we have like a blog in our website that talks about like the different research that is done with zebrafish, that talks about researchers that use the zebrafish model or like other model organisms. So we publish one article per month. So every month at the end of the month, you will get like some new discoveries using zebrafish. We're very happy if you actually, like you guys or whoever is listening to this, collaborate on some articles. Like we can publish about the research that you're doing. So it would be pretty cool to work on like joint articles. Actually, we did that. It's one of my favorite article series that we've done like last month of March. Every week we publish the work of a female researcher that has been done in zebrafish, and then we go to talk with some of the researchers that were doing that job, and it was super cool. So we're very happy to spread the word about the cool things that you guys are doing. We also have a newsletter, so don't forget to sign up for that. So you will get the article straight in your mailbox. So yeah, that's a bit the media that we want to share with the people. Like we want to create things that will be like useful for you. So happy if you have like hot topics you would like to learn about.
That's great. We'll make sure that we share at the end just kind of different ways folks can get a hold of you. And we'll make sure that we share that in our show notes, all that kind of stuff. So we want to chat a little bit about kind of future directions for both your company as well as the EggSorter. What kind of things are on the horizon that you want to talk about or able to talk about to share with us on the podcast? Yeah.
So we have many ideas. I will be happy to discuss with you afterwards the ideas that you would like us to do. Like kind of getting crazy and be like, okay, what would be your dream EggSorter? How would it look like? Or like any problems that you might have in the facilities. We might just build in a couple of years a machine for that. But yeah, for us, it's more like adapting the EggSorter to the applications that you will be interested in, like working with this AI, all the different possibilities that are there. And also something new and exciting is like adapting the EggSorter for the aquaculture industry. So
cool.
species that are being used in aquaculture. So yeah, but I will be very happy to discuss with you now, like what would be your ideas for us? Like, okay, what do you want us to do, guys?
You guys have any thoughts? I got to think about this.
Man, you really caught me off guard.
Sorry.
I mean, I was thinking like, I was kind of like having like a fun thought in my mind, which is just kind of hilarious, which is like the idea that like, if we could get the EggSorter to just learn how to feed the fish and walk around, and then like basically our jobs would just be obsolete.
Can we have EggSorter PIs so I don't have to deal with humans anymore, please? I make the eggs, I put them in the EggSorter, they spit out the eggs, and I put them on the system, and I've eliminated humans in my day-to-day. No, I'm joking. I love you, PIs. This is great. Amber?
I think for my purposes, a lot of the times, again, when I was talking about reviewing protocols and the PIs tell us like how many animals they want to use for their study, a way to connect that to the EggSorter. So maybe some other type of program that is connected to the EggSorter where it tells you real time, like, you know, hey, because I think that's also kind of a thing I used to deal with in my previous positions is like writing down a number. But then, of course, that number can change over time. So right now that I know the EggSorter can only do like, not larval, can only do embryos. But, you know, if you guys continue to make your way towards expanding that, I think that would just be amazing. So that way we can, you know, hold people accountable to like, you know, the animals that they're using for their projects, and we can reduce waste. And then, yeah, also keep the fish happy too. Makes me sad when I see all those old fish on the system not being used.
The dusty old fish. Yeah. And I think probably to add to that a little bit, now this is not necessarily EggSorter related specifically. This is more kind of where I'd like to see AI go in the future. Not that I want to give up my job as an animal health professional, but, you know, I've got 30,000 adult broodstock fish in my facility, and it falls on myself and my incredible team to like look at every single one of those fish every day. And we're not just looking at the fish as individuals, we look at the tank holistically, right? So we're looking at that whole tank, and we're seeing how the fish interact with the tank, how they interact with each other, you know, what their body condition looks like, all of those kind of things, where there has to be a way to automate that. Do you know what I mean? Where, you know, there can be a way where someone is being able to generate a report based on, you know, AI imagery of these animals, tank by tank, that can say, hey, have a look at these tanks. These fish are abnormal or maybe abnormal. The same thing too, sometimes when we have experimental animals, and again, this is outside of that embryological stage where people have specific phenotypes that are abnormal that they're looking for developing after a certain number of days post-fertilization. So these can be fish with neurological things where, you know, an AI software could detect that these fish are beginning to swim abnormally. You know, we have fish that maybe have a seizure phenotype, and having to have someone come down at a certain point in time and look at that tank rather than, you know, having a eye digital whatsoever on that tank and say, hey, these fish are starting to exhibit that phenotype, you need to sample them or whatever. So for me, you know, the EggSorter thing is incredible. And I just would like to see things like that extended to, you know, more automation in the fish facility, as far as, you know, our goal always is to not have to find fish dead, right? Like we don't like finding our broodstock dead. That's not the goal. We always want to be able to intervene and find a fish that is, you know, generally predictably declining in health, and then we can intervene and make sure they have a humane death. And, you know, when you've got 30,000 fish, you're still going to find a couple dead every day because we're humans and our eyes are fallible. So, and even something as simple as a tank up high versus a tank down low versus a tank at eye level and how well they get observed. Whereas, you know, AI can be non-discriminatory when it comes to those things. So that's my soapbox on AI. and fish. So get to work on that, please and thank you. Somebody.
I think it's a very good point. And to be honest, I don't remember who told me this, but I think somebody is trying to do that, like to have some cameras that will screen the tanks and have some AI that actually will check how the fish are. And I think it goes back to this point of automation, and maybe to comment on what Amber mentioned about like kind of mixing all this information together, right? Like shouldn't be like automation for this and automation for that, and then getting the data and then not knowing how to combine that together to make the life of the fish easier. So there should be a way to combine all this data, and it's where like a program that will take all of these together, because that's the thing. It's like with the EggSorter, we can track how many embryos we've placed, so you can know more or less how many fish you have in there and how many fish you have to take care of. And then this could be added into this AI that will take a look at the tank. Like the AI will know already, like there are 50 fish. I mean, 50 fish might be like maybe a lot, but it's like there are like X number of fish. So I know a bit what to expect, or I know what to look for. So should be a workflow where like all these automated devices communicate together, so that then the value that it brings will be like much higher than if they were like just a box and then a box and then a box and then. So this is where like all these AI and data comes into play, and I think it's great, and I really want to see the future in that way, like try to mix everything together so that we try. Well, Brian likes to say, like, I like to save the world. I don't know, that's big words, but at least try to make it a bit better if possible.
And how about you? Like, is there anything really fun that you would like to develop? You know, if you had infinite R&D money, right? Is there something that is like a project you want to do? If you're willing to share.
$6 billion. Come on.
I mean, we have had like plenty of fun ideas in the office, like this EggSorter that is transparent and has LEDs all over and makes like one beep every time there's an egg that has been sorted.
That won't get annoying at all.
Get an RGB set up, like a gaming computer, have an RGB, all the fans on it that are all constantly pulsing a rainbow of colors, like my keyboard is right now in front of me. That's pretty funny. That's cool. Sign me up. That would be like a special limited edition.
I like it.
Exactly.
Well, we probably should get ready to wrap up, unless there's anything else that you wanted to add, Anna.
I think we're good. I think it's been a great talk.
Sure. So why don't you share with us a couple of the ways that we can check out the EggSorter, and that folks can check out Bionomous, and let us know the best ways to check out your website or your socials, and we'll make sure we share those as well on our show notes for the episode.
Yeah, so for the website, it's bionomous.ch. So it's not .com as we would expect, because we're in Switzerland, so we keep it local. And then if you want to reach out to us, you can write to us at info@bionomous.ch. And you can find us on Twitter. You can find us, well, I would say X, not Twitter. Twitter is a bit old. So. And you can find us on LinkedIn too. So you can type Bionomous, and it's easy to reach out to us. And please don't forget the newsletter.
Yeah, absolutely.
Yes.
We'll make sure that all of those things are shared on our socials around when the episode comes out, beforehand and a week after. So we'll make sure it's in our little email newsletter as well, to show all the ways to find you guys and find out what you do. So how about any like conferences or meetings or symposia? Are there any of those that you guys are going to be at? Because I think maybe we may have crossed paths the odd time, but how about as Bionomous, are you guys going to be anywhere that we should be aware of in the next year or so?
Good question. So yes, we will be present in different like Zero Fees meetings. So in the US, we will be in the Mid-Atlantic meeting and the Midwest meeting. Then in Europe, we have a couple of those. Also, like we will be present in a workshop in EggSorter. We'll be present, of course, in the Swiss Zero Fees meeting. So yeah, you will be able to see us in person there, and we will present the EggSorter in live, so you can see how it looks like in the flesh. So yeah, looking forward to meeting all of you there.
Shall we wrap it up, Josh? Do you want to wrap it up?
Absolutely. Thank you so much, Anna and Bionomous, for sponsoring this episode. It's been a true pleasure, and it's a pleasure getting to meet you guys in person. I was lucky that they got to visit in New York, so exciting.
Very cool.
All right, wrap up time. Thanks for listening to Getting Fishy with It. You can find our website with show notes at gettingfishypod.substack.com. You can find us on Twitter at gettingfishypod and on Instagram at gettingfishypod. You can also find us on Facebook and LinkedIn by searching for Getting Fishy with It. If you want to drop us an email, you can send your complaints, questions, or adulations to gettingfishypod@gmail.com. Thank you so much for listening. Our theme music is Best Time by FASSOUNDS, and our audio is edited by the arduously hardworking audio drop enthusiast, Amber Park Chiodini. We've been getting fishy with it, so keep schooling, my friends, because knowledge is power.